I'm ready to go. I can start now? Okay, cool. Well, thank you everybody for coming on to the final talk of the Biocord Winter Conference. Grateful to the Biocord admins for saving the best for last. Kidding, just kidding, of course. Give a brief introduction of myself. My name is Yatri. I run a small server known as the Organic Chemistry Server. I know, clever name. And what we do over there is not only help people understand the basics of organic chemistry at the undergraduate level, but we explore even more advanced topics, which includes some applications of organic chemistry into biology, and that's basically going to be the basically what I'm going to be talking about today. And hopefully, my goal is to basically show you biology through the lens of an organic chemist, because that's basically what my background is. And I do have a little bit of background in pharmacology from my undergraduate studies, but hopefully I can show through chemistry how and why certain biological phenomena occur. And then, as indicated by my slides here, my cover slide, hopefully you like it. I personally like it. There's a lot that's going to be discussed, and the mechanisms are definitely going to be dived into. So without further ado, we're going to start. So there's going to be two parts to my talk. The first part is going to deal with review and explanation of certain basic chemical concepts, and then we're going to we're going to basically zoom in on proteins, break it down into to a deeper level, and then explore some certain enzymatic mechanisms that are in biology. And then in the second part, what we're going to do is talk about applications of bioorganic chemistry, specifically with drug discovery and drug development, as well as talk about the interesting applications that have been existing over the last last decade or so. So let's start with part one. And I want to start off by this question over here on the top, which is, what's the huge deal with biology anyway? And, this is something that I ask myself, for instance, when I've taken introductory biology courses, whether it be intro to molecular biology or intro to physiology or intro to ecology. And I always want, I'm learning so much. I'm trying to process so many facts that's being told in the lecture, and then I still wonder to myself, maybe because of my inquisitive nature, why is that the case? And I was trying to think about it, the answer to this question, and I remember that one of my good friends, he's currently at UCSF, does studying, he's doing a biophysics PhD. He sent me a really neat article from Nature from 2011, and it basically kind of it basically answers this question in a very nice way. And I remember at the top of the article, it shows this. It's all about the structure. And basically, in this article, you have this famous biologist, Brian Kobilka, who basically solved the crystal structure for this G protein coupled receptor called human beta 2 adrenoreceptor. And, he was talking about how, the main thing and the main motivation for studying and figuring out this is that, figuring out the molecular structures and whatnot. And now this is something, for instance, that you guys are well familiar with, the protein scene, for instance, alpha helices and beta pleated sheets. But, digging deep down into the molecular level and constructing everything together in a three-dimensional view to really have that true representation of the protein. And one of the quotes that he says, which I think was quite elegant, is that that's what makes scientists go. That moment when we know we found a new continent. I definitely agree with that. And that basically summarizes science, where no matter what idea you may have or what you're trying to study and explore, it could lead you to a path that you think is intended, or even lead to a totally new path that you never thought would actually happen. And I think through studying, biology and understanding the various ways in which proteins and enzymes can really function, can give rise to new paths. So that's really, really exciting. So before we get into how and why this looks like this, I'm gonna have to give you guys a quick primer on organic chemistry. Now, I'm well aware that the majority of you are probably don't have as strong of an organic chemistry background, which is totally fine. And I know organic chemistry spans 2 semesters, but hopefully within the next 5 to 10 minutes, I'll be able to summarize what is the essentials of organic chemistry and the basics that you'll need to probably understand when I'm going to be talking in my future slides. So the first thing we're going to do is review basic chemistry concept known as the octet rule. So you have atoms attached to each other, which are molecules, and for each atom, you want to have 8 electrons around it. That's the golden number. And there are exceptions, of course, if you have hydrogen that's attached to your molecule, you want 2 electrons around it. It's sometimes called that the duet rule. And then for elements third row and beyond, for instance, phosphorus and sulfur, which are common atoms that you see throughout, biology, they could adapt multiple bonds. So, for instance, if you can think, the phosphate group is a common biological group that has 5 bonds to it, and every bond consists of 2 electrons, so it has 10 electrons around that phosphorus atom. And the reason why it can adapt that much, so many electrons, is due to what's called hypervalency. And the easiest way to explain hypervalency is in elements third row and beyond, it's going to have these empty d orbital shells, which can take upon extra electrons, and in doing so is able to take around a higher electron configuration. So that's what hypervalency means. So that's the octet rule in a nutshell. And now we have to go into how chemical structures are written. So this is something you probably are well familiar with seeing in your general chemistry classes, the common Lewis dot structure, where you have the bonds being drawn and the lone pairs being assembled in this fashion. And for a small molecule like this is fine for drawing it out. But if we go back to the previous slide, this is if you look at our proteins, really, really huge molecules. If you try to draw the Lewis dot structure for that, it would be really complicated. So August Kekulé came up with a way to basically draw molecules in a more neater fashion. And what you can notice is the following. For instance, here I'm not sure, you don't have to necessarily show the lone pairs of the chlorine atoms. Also, notice that now we don't show the carbons and hydrogens explicitly. So, for instance, here would be a carbon atom, here would be a carbon atom, and here would be a carbon atom. So, at every point, you're going to have carbon atoms. And then you assume at every point where there's carbon atoms, if there's unless there's 4 bonds, the rest of the bonds are going to come from hydrogens. So you're going to have 3 hydrogens here on this end, and then 3 hydrogens here on that end. And then finally, the great thing about Kekulé structures is that it represents the 3-dimensionality of a molecule. So for instance, here I'm illustrating the 3-dimensionality through wedges and dashes. So imagine if you had some sort of 3D glass and you put it on, you can imagine that the wedged bond is kind of facing, towards you, and then the dash bond is facing away from you. So that's the 3-dimensionality that we're talking about. And speaking of three dimensions, we have this value, which is the steric number, and the steric number around the central atom determines the molecular geometry. And these are the most common geometries that you're probably going to be dealing with. And I've steric number defined here, which is the number of bonding pairs plus the number of lone pairs. One thing to keep in mind is that, bonding pairs, if you could have a single bond, a double bond, and a triple bond, each are treated individually as one bonding pair. So I'll briefly go over like one example. So if you have a steric number of 2, you're going to have a linear geometry. Common example, everybody's probably well familiar with this molecule, carbon dioxide. If you look at the steric number, it's 2 because of the fact that you have this double bond over here, which counts as one bonding pair, and then you have the other double bond, which counts as another bonding pair, resulting in having 2 bonding pairs, hence a steric number 2, and as a result, the angle is going to be 180 degrees. Then if you have a steric number of 3, you could have trigonal planar geometry, have an angle of 120 degrees, and then finally, you could have, if you have a steric number of 4, you could have tetrahedral geometry, which has a calculated bi-angle of around 109.5 degrees. And then there's other steric numbers. You could have 5, 6 that represent other geometries, but these are the most relevant and the most common ones that you'll probably encounter when looking at biological molecules. So the next part I want to talk about, probably the last slide with this primer, is discussing the concept of resonance. So molecules have a specific electron configuration where the molecules can basically, not the molecules, but essentially the electrons can move around and basically shuttle around. And this is what we call resonance structures. So you can think of resonance structures as snapshots of the molecule, and it shows this delocalization that can occur. And to draw resonance structures, you have to understand what curved arrows are. And then as I'm showing here, curved arrows are used to represent electron movement. So if you were to talk about how that works, your arrow always starts from where the electrons are located. It's going to be either a bond or a lone pair. It never starts from an atom. And then the arrows can end either on another bond to form a double or triple bond. It can end on another on an atom to form a new bond, break a bond, or put the electrons elsewhere. You could put a lone pair on that atom, for instance. And then finally, if you put them in between atoms, you could form a new single bond. And this will be a little bit clearer right now because what we're going to do is a quick example of drawing resonance structures for this compound known as 2-pyrone. So this is the structure over here. The first thing I'm going to do is, if you look closely at this bond, which is the carbonyl bond, this is going to be, it's going to be a very key bond that you're going to be seeing all throughout today's lecture. Definitely pay close attention to this bond. If you were to take the electrons and move it onto the oxygen, you get your first resonance structure. And it looks like this, where you have a negative charge on the oxygen and you have a positive charge on the carbon. And these are known as formal charges, and I have the formula for formal charge over here. So I'm going to do this quick example for you guys to understand why the formal charges are the way they are, and get out my pen. So let's look at oxygen, for instance. Oxygen has a total of 6 valence electrons, right? So if we take that and we try to sum up the total of bonding and non-bonding electrons, what you can see here, I'm going to draw it out, hopefully you guys can see it, is that you have 3 lone pairs. Sorry if my drawings are not the best. I'm not an artist. But basically here what you see is, it has 3 lone pairs, and for every lone pair, you're basically going to have 2 electrons. So that's going to be 6 non-bonding electrons. And then as you can see right over here, you have 1 single bond. So we're going to add a 1 here. And then recall from PEMDAS, you do whatever it's in the parentheses first. So really you have now 6 minus 7. And as a result, the formal charge is going to be -1, hence why the oxygen bears a negative charge. So I'm going to clear this real quick. And now I'm going to quickly do the formal charge for this carbon over here. So carbon has 4 valence electrons. Here it's a little bit easier to see because primarily we're not dealing with any lone pairs. So we know that the number of non-bonding electrons is going to be 0. But as you can see, carbon has 3 bonds to it currently. So the total number of bonds is 3. So 4 minus 3 is equal to +1. Now, one thing to keep in mind is that this resonance structure isn't really stable, primarily due to the fact of the carbon that bears the positive charge, which we call carbocation. Now, remember that with the octet rule, you want to have 8 electrons surrounding it. Now, you only have 3 bonds here. And remember, I mentioned earlier that for every bond, you're going to have 2 electrons. So as a result, you only have 6 electrons around that carbon. And as a consequence, that basically means that the carbon doesn't satisfy its octet. And this is going to be a little bit important later on because it turns out that the reactivity of that carbonyl bond with the CO double bond is because of the fact that through this resonance structure, you have sort of like a partial positive charge on the carbon. So that's something to keep in mind. Okay, but hopefully you guys were able to follow that simple example. So let me get back my laser pointer, and we'll continue drawing the rest of the resonance structures. So here it will be a little bit easier to follow. So for instance, if you were to move this double bond over here, what you're noticing is that you're also subsequently moving the positive charge down to the bottom. And then basically you're going to notice this pattern where for every double bond you're moving, you're also going to be shifting that positive charge. And it's going to work out the same way when calculating the formal charges. You're going to only have 3 bonds to that carbon, and that's why it's going to have a positive charge. Let me take a quick drink of water for a second. Okay, so we have this resonance structure, and I drew out one of the lone pairs now for this oxygen. If we swing those electrons like so, we can now end up at this resonance structure. And this is one of the major contributing resonance structures. And the reason why I say it's major because of the fact that if you look at all of the atoms, each atom has their octet being satisfied. And that's typically a good indication of having a very stable resonance structure. And likewise, if you were to just move the double bonds all around like so, in basically a clockwise direction, you can end up at this similar resonance structure. And again, the octet is satisfied for all atoms, so this would also be another major resonance contributor. So note that no single bonds are broken in this process. If you do, you're basically fragmenting and changing the whole properties of the molecule. So notice one thing that's consistent with all these resonance structures is the fact that you have you have sort of like a hexagon type backbone. So that's being maintained all throughout. And then finally, if you were to combine the most contributing resonance structures, you could end up having a resonance hybrid. And note that in the case of this resonance hybrid, and also with all the resonance structures that we've drawn, all the charges equal 0. So that's also something to keep in mind. You want the charges on your resonance structures, the sum of that to reflect the starting molecule that you had. So for instance, since 2-pyrone is stable, itself in this form. When you draw the resonance structures, each of these the total charge is 0 in each. So that's so I've now given a clear example of how resonance structures are drawn, and then based on these arrows, that's going to tie into mechanisms and describing certain transformations of various reactions. So hopefully that was easy to follow. I know I probably sped through that a little too quickly, but hopefully this was easy to process. So now we're going to move on to talking back to proteins. Talking about proteins, I'm going to break a protein down, and the biggest thing to note is that structure dictates function. So I'm going to start off with this tertiary structure. This is a mouse prion protein, has this PDB code 1AG2. And with the tertiary structure basically describes the specific folding and conformation that it takes, specifically with these secondary structures. And if you were to zoom in, you could see more as to why these secondary structures arise. So, and the primary thing that's consistent between both of these secondary structures is the fact that you have this hydrogen bonding that's occurring. And depending on the type of hydrogen bonding that's occurring, that's going to either lead to beta pleated sheet or you're going to lead to alpha helix. Now, if we want to dive in even deeper to understand why we arise to the secondary structures, we have to look at the amino acid sequence, also known as the primary structure, which is shown over here. And speaking of amino acids, that's what we're going to be talking about next. And amino acids are the building blocks of proteins. And this is the general structure of an amino acid. So you have the reason why it's called an amino acid is because you have an amine group and you have a carboxylic acid group. Okay. And remember that amino acids exist within our body. So at physiological pH, it actually turns into a different form, which we call Zwitter ion. The reason why it's called a Zwitter ion is because of the fact that the molecule has charged parts, but the overall net charge is equal to 0. And the reason why certain parts are charged are because of this value pKa known as the acid dissociation constant. And its a relation to the pH alters whether or not you have either gain of a hydrogen or loss of a hydrogen. So to briefly explain, if you take the pKa of a carboxylic acid group, it's generally around 2.3. Whereas if you take the pKa of the amine group, it's generally around 9. Now, the idea is the following. If the pH is greater than the pKa, then the group is going to be deprotonated or it's going to lose a hydrogen. And that's why in this case, you have the carboxylate where you're missing one hydrogen. Also, if the pH is less than the pKa, then it's going to be protonated. That group's going to be protonated or have an extra hydrogen. In the case here of the amine group, you're having an extra hydrogen, hence why it's NH3+. So that's the relationship between pH and pKa. And as you could also notice, there's an R group attached here, and there could be 20 different R groups that can exist. And I found this diagram online. I think that best summarizes it. And based on the specific R groups, you can have certain functionalities. So you can have hydrophobic R groups. You could have polar R groups, and then you could have like electrically charged R groups, etc. So now the next thing to talk about is how the amino acids come together. So if I go back, notice that the amino acids here are connected by this bond. And this bond is known as a peptide bond or an amide bond. And amide bonds basically string up these amino acids. So this is the typical structure. What I'm going to do now is draw a resonance structure of this. So if we take the lone pair on this nitrogen and move it to the left, then the double bond from the carbonyl is going to move up, and you're going to have the subsequent resonance structure. Now, the significance of this is that the carbon-nitrogen bond, which originally is a single bond, actually turns out to have partial double bond character. So one thing to note is that single bonds have this property of being able to freely rotate. But because of the fact that you have this resonance structure, double bonds are more rigid. So that means that that single bond can't actually rotate as well. And that's something that basically describes the properties of an amide that also give rise to why, for instance, you have alpha helixes and beta pleated sheets. Now, there's one interesting enzyme that I found online that basically exploits how this bond is basically conformed, and it deals with this enzyme known as proteolysimerase. So proteolysimerase alters the amide bond configuration. So For instance, this is the proline amino acid, and currently in the trans configuration. I'll briefly explain why it's called trans in just a second. But if you were to take this enzyme, what you could end up doing is normally this carbon nitrogen bond, which is really hard to rotate, can end up being rotated, and you end up having this cis conformation. Now, if we apply the concept of resonance that we've shown over here, we can draw resonance structures for each of these. And it's a little bit easier to see why it's called trans and why it's called cis. So for instance, it's called trans over here because these bolded groups are pointing in opposite directions. You have this being pointed downward, and then you have this other group being pointed upward. Now, in the cis resonance form, you can see that both bolded groups are being pointed up and in the same direction. So that explains why that's called cis. Now, what's interesting is that this mechanism is not well understood, but there is one proposal as to how this mechanism proceeds. So I'm going to get out my pen again. And what biochemists have proposed is that there's a small chance that the nitrogen ends up getting protonated. Now, if this gets protonated, this basically means you don't have a lone pair in the nitrogen. And if you don't have a lone pair in the nitrogen, you cannot do this resonance. This resonance is not able to occur, and then now the carbon nitrogen bond acts more freely as a single bond and is able to rotate. So if that's protonated, the enzyme helps to rotate that. It'll still be protonated, and then basically to end the mechanism, you would just take off the hydrogen, put back on the lone pair, but it'll be stuck in that cis conformation because through resonance, it will have more of a rigid bond and it will stay in that conformation. So that I found was quite interesting. So that's basically that's the basics of the amide bond. Now, we're going to talk about one reaction of the amide bond, which involves hydrolysis. And this is the general reaction scheme of the hydrolysis reaction. The reason why it's called hydrolysis is because hydro involves water. So for instance, here you have the hydronium ion, which is just basically dilute acid, H3O plus. And what happens is if you react the amide with H3O plus, you're going to end up with two products, the carboxylic acid and an amine. Now, the easiest way to probably conceptualize this is imagine you had some scissors and you were to basically cleave or cut off this carbon-nitrogen bond, and then in the place of the nitrogen, you basically stick on an OH. That's what leads to the formation of the carboxylic acid and the amine. Now, we're going to go over the first mechanism of this talk, and specifically the mechanism for the hydrolysis of an amide. And I'll go a little bit slow, so hopefully everybody can follow. So the first step that's going to occur is you're going to have an acid-base reaction between the amide and the hydronium. And the easiest way to rationalize this is if you to take one of the lone pairs of the oxygen that's part of the CO carbonyl bond, and it's going to pick up a hydrogen, and then the oxygen-hydrogen bond from the hydronium is going to go onto the oxygen, and that's going to result in a protonated form of the amide, and you free up water. Now, because it's now this amide is protonated, it could basically function as what we call a good electrophile, meaning that it wants to be attacked very well. And what attacks it is the water, which acts as a good nucleophile, or what I'll call a quote-unquote attacker. So what happens is you take one of the lone pairs on the oxygen, you attack that carbon, then the electrons move up to the oxygen, and what happens is now the oxygen is going to have it's going to be stable, and now the positive charge is going to move on to this water molecule. Now, the same logic applies. You want to try to stabilize the oxygen, so now you can use the nitrogen here to grab the hydrogen. That oxygen-hydrogen bond is going to break to stabilize that oxygen, and now the nitrogen has a positive charge. Now, finally, what you can do is have one of the lone pairs from either one of the OH groups swing down, and then finally you're going to kick off this amine group, and you're going to result in this, where you have the free amine, but you have now this protonated carboxylic acid. And then finally, the protonated carboxylic acid can lose its proton either by water being picked up or even the amine picking it up. And what you have as a result is the free carboxylic acid. Now, that's the whole logistics of a mechanism. The best way I try to rationalize it is think about like solving a Rubik's Cube, right? It's all mumbled and jumbled and whatnot, and you have to basically, keep, turning it around and around until you finally get to solving it where all the sides have basically the same color. Now, it's kind of analogous to mechanisms. You kind of have to play around with electron movement keep on attacking, shifting electrons and hydrogens and all that other stuff until you finally get to the final product. So I think that's like probably the best way to describe mechanisms. It's similar to a Rubik's Cube where you try to manipulate it and play around and finally get to the end result. Okay, so now hopefully that's clear to you guys, and we're going to look at an analogous reaction that happens within biological systems, specifically cysteine protease that catalyzes amide hydrolysis. So cysteine proteases are really unique because of the fact that they're found in fruits such as papaya and pineapple, etc. And as I mentioned before, this is how this basically an enzyme would catalyze the hydrolysis of an amide bond. So the key residues that are amino acid residues that are going to be involved include the cysteine 25 and the histidine 159. But similar electron movement is going to occur. Now, in this case, the first thing that's going to happen is the negative charge on the sulfur is going to attack the carbon of the carbonyl bond, and you're going to push up the electrons to the oxygen. As a result, you form this intermediate known as the enzyme substrate complex because now the enzyme is bound to the peptide essentially. Now, oxygen has a negative charge. Oxygen is electronegative, so it could stabilize that well, but the end goal is to always keep trying to stabilize stuff. So what can happen is you can have the electron swing down from the oxygen, and then you could have the carbon-nitrogen bond pick up this proton from the histidine, and then the electrons can go on to the nitrogen, and you could end up with this falling intermediate. Now, I kind of want to go back a little bit, and I'm going to get out my pen again. And what I want to discuss is, what if the histidine wasn't there, for instance? So imagine the histidine wasn't there, and now you put the electrons simply on the nitrogen. What you end up having as a result is the following intermediate. You have a negative charge, and then this is basically the nitrogen, the free amine group of the peptide. Now, the issue here is the fact that nitrogen isn't a large enough atom, and it's also not electronegative enough to stabilize that negative charge. So in fact, if you were to do this reaction outside of the biological system, you typically wouldn't have the amine being kicked off as a leaving group for those reasons. But fortunately, what we notice is the fact that because of the fact that this protonated histidine is in close proximity, it could immediately just pick up a proton, and then as a result, now the amine that was kicked off is rather stable, and it doesn't bear any negative charges. So now moving along, since again we're dealing with hydrolysis reactions, we're going to need water, and of course, since we're dealing with an enzyme, it's going to be in aqueous media by default. Now, typically, water is not a good attacker. So the way we can make it a better attacker is to utilize the histidine residue. What it's going to do is it's going to grab the proton, and then subsequently this oxygen and hydrogen, the electrons from the oxygen hydrogen bond are going to go and attack now this carbon again and pushing up the electrons. And something that you're probably noticing right away is this pattern of attack and kick off, an attack, and of course, if you guess correctly, you're going to kick off again, and you're going to finally get the free carboxylic acid, but you're also going to regenerate the cysteine 25 residue. Now, one thing you might be wondering is why the sulfur can basically bear a negative charge as opposed to the nitrogen. Well, based on the position on the periodic table, because of the location of sulfur, it's a really, really large atom, and large atoms are very good at handling negative charges and stabilizing it pretty well. So for those reasons, that's why sulfur can basically be kicked off as a good leaving group and exist having a negative charge. So as you can see, it follows a similar mechanism to the previous slide, where again, we're just breaking that carbon-nitrogen bond of the amide, and you're basically forming a carboxylic acid, and you're forming an amine. So hopefully, again, this mechanism is relatively straightforward to follow. Okay, now we're going to shift, not necessarily shift gears, but we're going to look at a different type of reaction, which is known as the decarboxylation reaction. Now, the decarboxylation reaction is pretty self-explanatory. You're eliminating carbon dioxide from the system. And again, an easy way to think about it is if you were to take scissors, kind of just cleave this specific bond, you see that this portion, this carboxylic acid portion, looks quite similar to CO2, and then through the mechanism, which we're going to see right now, basically eliminates itself as carbon dioxide. So you have this molecule over here. And the thing to keep in mind, by the way, it's called a beta keto acid. And the reason why it's called a beta keto acid is because if you were to take this carbon as your reference point, one carbon away from that is going to be called alpha. The second carbon away is going to be called beta. This is a ketone, so it's keto, hence beta, keto, and acid because you have a carboxylic acid. So that's the sort of naming that goes along with that. And this molecule is unique because, what, as I mentioned before, remember single bonds have this intrinsic property of being able to rotate. So if you were to rotate this bond 180 degrees, you would end up with this conformation. And one thing you could kind of notice is that based off of the proximity of this hydrogen and oxygen, you could have this sort of hydrogen bonding that occurs, and you could kind of have like this pseudo six-membered ring, sort of a pseudo hexagon type structure. Now, that along with the fact that you have heat will drive basically the elimination. And note that I drew the arrows here at the same time, so it's called what we call a concerted process. And what happens as a result is you're going to eliminate carbon dioxide, but you're going to form this compound known as an enol. The reason why it's called an enol is because of the fact that you have a double bond, so the ene, and then the alcohol because you have an OH, so enol. And what you could do is if you treat this with some acid, indicated here by H+, you can revert the enol back to the ketone form. So the one thing to keep in mind is, along with hydrogen bonding, as I mentioned, heat is the driving force in order to eliminate carbon dioxide. Now, I showed you the case for beta keto acid, but what if you have an alpha keto acid? And we're going to look at that example right now. So this is an enzyme that's very common, pyruvate dehydrogenase complex. It not only catalyzes the decarboxylation of oxaloacetate, which is our alpha keto acid, but it also is involved with making acetyl-CoA, which feeds into the citric acid cycle or the TCA cycle or the Krebs cycle, whatever name you like best, feeds into that cycle. And it's a complex because there are three parts. You have E1, E2, and E3, each with their own individual functions. So I'm going to start with the mechanism of E1. You're going to need is so what's going to happen is you're going to have the loss of this hydrogen over here. You're going to have the subsequent bond formations, which eventually leads to the deprotonation or the loss of the hydrogen of the aspartate. And what you're going to have as a result is this intermediate over here. Now, the aspartate can reprotonate itself by grabbing the hydrogen. And one thing to keep in mind now is that the carbon-nitrogen double bond can now pick up this hydrogen over here. The electrons go into the oxygen, and you end up with this carbene intermediate. Now, the reason why it's called carbene is the following. So, going back to resonance structures, if you were to move the double bond over to that nitrogen, what you would have as a result, and I'm only going to focus on the five-membered ring, you lose the charge on the nitrogen, you would have the sulfur. What you have as a result, you have no charge on the species. But what's weird is the fact that you have a lone pair on that carbon. Now, what's even more weird is the fact that with that carbon, it actually doesn't have a stable octet because there's only 6 electrons around it. So these sort of species that have a lone pair but also don't satisfy the octet rule are known as carbenes. But I'm going to be mainly focusing on the bottom structure, which is more resonance stabilized, because as you can see here, there's a total of 8 electrons in this figure over here, in this structure over here, and because it has 8 electrons, it satisfies its octet. So let me clear this, and let me get back my laser pointer, and now we're going to use this to catalyze the decarboxylation. So continuing on with the mechanism, what's going to happen is you're going to have the negative charge attack the carbon, and then because something I should have mentioned before is the pyruvate dehydrogenase complex is found within the mitochondria. Mitochondria is always producing protons, so when this does the attack, this carbonyl bond is going to pick up a proton, and it's going to lead to this intermediate. Now, what's a little bit weird is that you're going to actually have the elimination of the carbon dioxide in this step. Now, you're probably wondering why this carbon anion intermediate is now stabilized. It's because of the fact that if you were to, and I'll get out my pen again to kind of illustrate the point, if you were to draw a resonance structure like so and move up the electrons, you would end up with having a double bond over on this over here, and then basically the nitrogen loses its charge. the intermediate is bears that resonance structure bears no charge. So that's kind of the driving force for forming this sort of carbanion intermediates, because it's resonance stabilized, because you still have that TPP attached to it. So now the next steps are going to be a little bit weird, but hopefully you can follow them. So you're going to lose a proton, but subsequently you're going to have the attack of this carbon ion, and then you're going to break off this carbon-carbon bond, and then the result of that is going to be reformation of this fourth. But then when this carbon ion attack is going to break and grab a hydrogen, and you're going to end up with this intermediate over here. This is what's called a thioester, and now you have a free thiol. So there's partial reduction, and that's something you've also heard in chemistry, oxidation and reduction. In the context of organic chemistry, reduction means you're gaining a hydrogen, whereas with oxidation, you're basically losing hydrogens. So there's some partial reduction here. Now, this intermediate now feeds into E2, where it's attacked by a coenzyme A molecule. So again, attacks it like in a certain fashion, and then like before we've seen, whenever this attacks, you basically have elimination. And again, since we're in the mitochondria, there's definitely proton source available. And then finally, you form acetyl-CoA, and you form the fully reduced fully reduced form here, which is known as a dithiol. Now, if you wanted to oxidize this back to the disulfide, that's where ER3 comes in. So what happens is through the reduction of FAD and NAD plus, this leads to the oxidation of the disulfide, which can then feed back to this step within E1. And then this could lead to forming this intermediate that feeds into E2, which leads to the synthesis of acetyl-CoA. So this is a whole cycle that occurs, and it keeps on occurring and occurring and occurring. So that's the whole mechanism behind the pyruvate dehydrogenase complex. I know, very complicated, but hopefully you're able to follow the arrows and things relatively easy to follow. And then one last thing I probably should have mentioned prior to is the fact that the reason why it's called pyrophosphate is that you have these two phosphate groups, and for simplicity's sake, I just labeled them as orange balls that contain the letter P. But you'll see how the structure actually looks like for those who don't know in a future slide. Okay, so the last slide we're going to get to, which will conclude the end of part 1, involves a unique reaction known as the Diels-Alder reaction and its enzyme called SpinF. So the general scheme for a Diels-Alder reaction includes the reaction of this compound known as a diene. And the reason why it's called a diene is because you have two double bonds. And then over here, you have what's known as a dienophile, which is just a simple double bond or alkene. And similar to the mechanism that you saw for decarboxylation, you're going to basically have this concerted process where all the electron movement occurs at the same time. And heat helps to drive this reaction outside of the biological system. And what you'll notice that's pretty unique is this thing here, which is called a transition state, which I haven't really talked about much. You can think of the transition state as the midway point between, the reactants and the products. And it also indicates what bonds are going to be formed and what bonds are going to be broken. So notice that in the case of the mechanism here, each of these double bonds are involved in electron movement. So they're going to have partial double bond character because they're going to turn into single bonds. Another thing to keep in note is that now the central single bond is going to turn into a double bond, so it also gains partial double bond character. And then finally, these two single bonds are going to form, so it has partial single bond properties. And then finally, this is what the product looks like. It's called a cyclohexene. The reason why it's called a cyclohexene is because cyclo means a ring, hex meaning 6, and then ene because it has one double bond. And hopefully you guys can see this. I've shown a bold orange that indicates the new single bonds that have been formed from this reaction. So that's the general schematic. Now, what's really unique is that in nature, this enzyme SpinF, which is found in this bacteria known as Saccharopolyspora spinosa, I know I butchered the name, sorry about that. This actually catalyzes the Diels-Alder reaction. And interestingly enough, this was the first enzyme identified to catalyze this, and is only really identified 5 years ago in this Nature paper. So I'm going to talk about how it catalyzes this reaction. So this is one of the intermediates within the biosynthetic pathway. But what you'll notice right away that this motif looks very similar to the diene that's shown here. Resembles it quite well. And then, as you can see, situated right across is having that alkene, that dienophile double bond, similar to over here. And if you follow the electron movement, it's going to happen in a similar fashion. You're going to have basically this transition state, and it should be a little bit more clear as to see how you're going to form this new fused bicyclic, or I should say tricyclic system, rather. And then as a result, that you're going to form basically the cyclohexene ring. And then finally, further modifications leads to the formation of this insecticide called spinosyn A. Now, so the authors looked more into the binding pose of this enzyme, and what they noticed is the following. So, if you look at the hydroxyl groups over here, you notice that they're involved in hydrogen bonding. So, specifically, they're involved with hydrogen bonding with tyrosine 196, histidine 42, and I think glutamate. I think it's glutamine or glutamate. It's one of those. I think it's glutamine 148. Now, notice that hydrogen bonding is consistent prior to the cyclohexene formation and after. So that's something to also note when it comes to enzymatic reactions, is the fact that the molecule has to adapt a certain conformation, and that's usually facilitated through some hydrogen bonding or formation, as we saw earlier in the cysteine protease, like an enzyme substrate complex, in order to carry out the reaction. But in this case, simple hydrogen bonding is good enough to have the appropriate conformation where you can actually form the cyclohexene ring and then eventually lead to the formation of this insecticide. So that's, I found to be really, really fascinating and cool. Now, that marks the end of part one. I know it's a mouthful. I'm almost out of breath, but I should be able to make it to part two now, where we're going to dig a little bit deeper into the basics of drug development, drug discovery, and seeing some unique examples regarding to that. So, I'm going to define a good amount of terms here and introduce the basics of drug design. So first, pharmacodynamics. What is that? It basically means what the drug does to the body. It's different from pharmacokinetics, which means what the body does to the drug, which is through ADME, or absorption, distribution, metabolism, and excretion. Here, basically, you're going to see how the drug interacts with certain enzymes and proteins to carry out its function. Now, drugs can be designed in the following ways. You can have a drug that mimics either the transition state or a binding mode, and this is an example of such a drug right here, SPFI 26, that mimics the binding of an Amide by maintaining the same amino acid, same interactions with amino acid residues. You could have a drug that resembles a metabolite or other biomolecules. So, for instance, here you have Abacavir, which is an NRTI used for treating HIV and AIDS. And then you could have a novel functional group that carries out some unique functionality or purpose. So, for instance, endiene drugs, which we'll get into, are common drugs that are used in chemotherapy. Now, drug discovery can be a very tedious process, and this is only just the beginning of what drug development really is. Really, the starting point is collecting compounds, doing some initial assays that eventually leads to promising leads. And then from there, optimization of those leads, designing them and synthesizes them, leads to studying those in new assays, and the cycle kind of repeats itself up until the point where you have a really, really strong candidate after the secondary assays, which can then be used for clinical trials. Now, while clinical candidates could be promising, results of clinical trials can be discouraging. And the reason why I say that is the following. If you think about, especially in the US, how drugs are approved by the Food and Drug Administration, or FDA, you have various clinical trials that has to go through phase 1, phase 2, phase 3, phase 4. And the thing that's unfortunate, I forgot the specific statistic, but a good amount of the drugs, the good amount of the clinical candidates that are used in phase 1 fail a lot. And that's very discouraging because you've worked all hard and you spend a lot of hours trying to get something that's a very potential therapeutic, only to realize that there's some huge issue in humans, and then you have to either, depending on the certain situation, unfortunately, you may have to abandon that project, or you may be able to find a workaround, and then you may be fortunate enough where it can actually make it further into the clinical trials. So that's something to keep in mind of. So even though you get through the whole preclinical studies, you're not really at the finish line. You're probably maybe 50% there, maybe even less. So that's something to keep in mind. So, but hey, this still motivates people to really make sure that we find therapeutics that work for us and to continue to improve treatments of various diseases. So this is not going to go away anytime soon. Now we're going to look at specific examples. So we're going to deal with transporter proteins and the endocannabinoid system. So transporter proteins are relatively straightforward in its name. Basically transport a molecule from one place to the other. Now, this is the endocannabinoid system. And I have the abbreviations over here, so I'm going to explain how the endocannabinoid system, it's involved a lot of various biological processes, but I'm going to be discussing how it directly connects to chronic pain and inflammation. So first, you're going to have this neurotransmitter called anandamide. It's going to diffuse through the cell membrane. And then fatty acid binding protein, which is the transporter protein, is going to be able to transport the anandamide over to this to the endoplasmic reticulum, where it's going to basically be hydrolyzed by this enzyme known as fatty acid amide hydrolase. So something you notice is that amide bonds aren't only limited to just being found in proteins. They can also be found in these neurotransmitters. And we've already talked about this before because hydrolase is similar to hydrolysis, and we've discussed before that whenever you hydrolyze an amide bond, you're going to have an amino acid and you're going to have an amine. So in this case, the free amine is going to be ethanolamine, and you're going to have the free carboxylic acid group, which is called arachidonic acid. Now, I remember watching Peter give his talk, Clockwork Show give his talk, and he's having a little bit of a difficulty pronouncing his name, but hopefully it's a little bit easier to pronounce arachidonic acid. So, and the reason why it's important, and Peter touched upon this in his presentation, is the fact that arachidonic acid is a substrate for the cyclooxygenase enzymes COX-1, COX-2, and then those lead to the synthesis of prostaglandins that feed into chronic pain and inflammation. So, how do you exploit the system to treat chronic pain? Well, what if you had a small molecule inhibitor that basically fits right into the pocket of the fatty acid binding protein? What happens as a result is that the anandamide can no longer be transported to the fatty acid amide hydrolase, meaning that the hydrolysis never happens. As a result, you have more anandamide now outside of the cell, and more of that is going to be able to bind to this receptor called the cannabinoid 1 receptor, and that's going to induce analgesia. Now, I was reading more on the paper on this, and the researchers, the way to describe this, interestingly, is that the way by exploiting the endocannabinoid system, it's a way of kind of activating the body's natural marijuana, and that's found that to be a very novel way to describe it. So, for instance, for those familiar with cannabis, THC, tetrahydrocannabidiol, is the active a psychoactive drug that's found within cannabis. And that also binds to CB1 receptors. And the same authors were able to illustrate that using THC can also be used. That's why, for instance, medical marijuana exists because of the fact that THC binds to CB1 and induces the same effect. So, likewise, anandamide can induce the same effect, and basically, you're trying to mimic the effects. You're basically mimicking the effects of THC, but within the human's body. So, I found it to be quite a novel and interesting explanation for that. Now, just like before, we talked about the a few slides ago about drug design. You order a bunch of compounds, go through the initial assays, and then you identify leads. The authors here identified 4 promising leads, the most notable one being this compound over here, which is called SPFI26. It's a 1-alpha trioxylic acid monoester. And the way that it works is that it inhibits the FABP5 protein through interactions of the carboxylate with arginine 129 and tyrosine 131, as shown here. And because they're able to find they're able to obtain a co-crystal structure that basically gives indication and confirmation that anandamide functions the same way, where it's going to interact with the same amino acid residue. So the main point of SPFI26 is that it mimics the binding pose of an anandamide, and that's why it gives rise to the formation of more anandamide. There's not going to be as much hydrolysis, and then that could bind to cannabinoid 1 receptors and basically lead to analgesia. So that I found to be quite unique. Now we're going to shift gears again to DNA synthesis. And this should, again, we're going to talk about another mechanism. So DNA synthesis is catalyzed by DNA polymerase. And the mechanism is thought to proceed through SN2, which is known as nucleophilic bimolecular substitution. So I'll explain what that means in layman's terms. You're basically going to have a nucleophile or an attacker, going to attack this carbon, and then you're going to displace what's known as the leaving group. It's going to adapt this transition state. And notice here that the steric number is 5, because if you count all the electron groups, you have 5. And even though I briefly had mentioned the geometry that was not shown in that table, when you have the steric number of 5, that means you're going to have trigonal bipyramidal geometry. So that's thought to be the transition state of this reaction. And as a result, you have this new compound over here where the nucleophile is attached. And notice that you basically have this inversion of this carbon. And if you were to flip it 180 degrees, basically what you'll notice that the hydrogen that's dashed is now going to become wedged, and then the hydrogen that's wedged is going to become dashed. But fortunately, you don't have to know too much about the three-dimensionality of this reaction. The only thing you should really take away is that you're basically replacing the leaving group with whatever the attacker is going to be. Now, in the case of DNA synthesis, we're going to see how that's done. So here is a typical example. You have two strands, DNA strand A and DNA strand B. DNA strand A has this 3' hydroxyl group, which is also seen over here for this guanosine. Guanosine is currently being hydrogen bonded or base paired to cytosine. And what's going to happen is a similar SN2 reaction where the electrons or the lone pair from the oxygen is going to attack the phosphorus, and then that's going to lead to kicking off this portion over here. So by the way, this is a triphosphate, and then once you kick this off, this is what's known as the pyrophosphate, which I had discussed previously with the pyruvate dehydrogenase complex slides. And the pyrophosphate is a very good leaving group because it's heavily resonance stabilized. And as a result, you end up having now now you have I think I drew this. I drew this correctly. Yeah, basically now DNA strand A is now attached to the guanosine. Now, similar thing can happen where if you attack the phosphorus and kick out the pyrophosphate, the same thing can occur, and this is going to keep on occurring until you build up DNA double strand. Now, you could exploit this by using this molecule over here called Abacavir. And Abacavir is structurally similar to guanidine. So what happens first is that Abacavir is transformed to carbavir triphosphate, which is its more active form. Now, one thing to keep in mind is the way that this molecule differs is if you look closely, you don't have an oxygen here, and then you lack also the hydroxyl groups here. So that's unique, and that's the function that it'll serve, as you'll see here. So now you have this enzyme, which is HIV-1 reverse transcriptase, which basically takes an RNA template and is synthesizing a new DNA strand. And a similar mechanism occurs. You have SN2 that occurs. And now this carbavir is now attached to the newly synthesized DNA strand. Now, one thing to keep in mind is the fact that because you lack that 3' hydroxyl group, it can no longer do the SN2 reaction. And as a result, this terminates DNA synthesis. And this is good because you don't want to keep on making that DNA strand from the HIV-1 reverse transcriptase. So making that subtle of a change can have such a big effect on saving somebody's life. And I think especially exploiting the simple mechanism like the SN2, quite novel. So I found that to be a really unique example. I'm going to deal with two more examples, and then we'll be done with the talk. So this is a enedione molecule called calicium myosin. This is the structure of it. It's very, very complicated. But the main thing I want you guys to focus is over here, which is known as the enedione. And the reason why it's called enedione is because you have one double bond and you have two triple bonds. And the mechanism of action involves two reactions, which we're going to briefly discuss right now, which is Michael addition and the Bergman cyclization. So let's first talk with the Bergman cyclization. If you take an enedione, heat it up, and treat it with some hydrogen source, you're going to be able to generate this benzene ring. So what happens? So it's different from this mechanism is different from the ones that you've seen, and that's because when you heat this molecule up, it actually proceeds through single electron movement, which we call also known as radical electron movement. So you're gonna have these fish hooks that indicate single electron movement. So if you have two electrons that join up together, you can imagine that's how we form the single bond. And then what happens is you have the other electrons moving onto the carbon atom. And what you form as a result is this biradical species, and this is very characteristic of the Bergman cyclization. And finally, if you treat it with some hydrogen source, you're going to be able to form the benzene ring. Now, the second reaction is the Michael addition, where if you have this specific molecule called the alpha beta unsaturated carbonyl, and I'll get out my pen again. The reason why it's called that is because the alpha and beta carbons that are involved in the double bond, which is also unsaturated, is close in proximity to the carbonyl group. Hence, why it's called alpha beta unsaturated carbonyl. So, let me clear the ink. Let me get back my pointer. What you notice is if you take this molecule and treat it with an attacker or a nucleophile, you have the disappearance of this double bond, but then also now you have the nucleophile attached to the outermost carbon. So the way that the mechanism works is that lone pairs attack this outermost carbon, you move the electrons eventually up to the oxygen, you form what's called an enolate, then the enolate collapses, and then you grab a proton, and then you eventually lead to the formation of that product. Now, one thing also to note is that the nucleophiles could be amines, dialates, etc. Now we can go into the mechanism of action for calcium myosin. So, a very unique functionality is this trisulfide. What's going to happen is you're going to have another attacker, could be whatever molecule that's within the body. It's going to attack that middle sulfur, and the electrons are going to be pushed onto the innermost sulfur atom. You're going to form a disulfide intermediate, and then you're going to have what's known here as a thiolate. Okay, and then as you can see here, this portion is basically an alpha beta unsaturated carbonyl, similar to what we saw with the Michael addition. And exactly that's what happens. You have a Michael addition. I'm not showing the enolate here, but basically an enolate forms, grabs a proton, and then it forms basically the final product. But the main thing to keep in mind is the fact that now you have the sulfur directly attached to this outermost carbon, and subsequently you have the disappearance of this double. Now, finally, we have the enedione left, and it could follow the same radical mechanism, and it could form this biradical species. Now, this biradical species, you imagine in a biological system, is gonna want to grab hydrogen, and then whatever it grabs the hydrogen from, it's gonna really affect it greatly. So, for instance, DNA serves as the hydrogen source, and then that eventually leads to DNA damage or apoptosis or cell death, and that's the thing that's important in the takeaway is the fact that through the Bergman cyclization and the formation of this biradical species, which is so well known to it, that is what eventually leads to DNA damage. So I think that was a really neat example of showing how you could have a specific structure that utilizes various reactions that eventually leads to the disruption of DNA when it comes to treating cancer. Now, the last slide is dealing with another chemotherapeutic, Taxol, and its drug delivery system. So you imagine a drug delivery system is some unique system that brings the drug closer to its target. So in this case, it's Taxol. Taxol or paclitaxel functions by binding to beta tubulin subunits of the alpha beta tubulin dimers of microtubules. Now, this leads to further stabilization, which prevents their disassembly. And if you're thinking about, mitosis and cell division, you want everything to be separated, but because it can't be separated, that's going to really mess up things. And as a result, that's going to lead to apoptosis around the late G2 mitotic phase of the cell cycle. Now, a novel drug delivery system has been developed that utilizes this common biomolecule known as biotin. Now, this is a schematic from the paper, and the thing to focus on is that you have the biotin drug conjugate, where you have the biotin molecule linked to the drug. And what's going to happen is the biotin is going to react with its receptor. That's going to lead to receptor-mediated endocytosis, where biotin drug conjugate is brought into the cytoplasm. This eventually fuses with the endosome, and in the endosome, you're going to have this molecule here known as glutathione that's going to basically cleave the linker, which leads to the free biotin and also the free drug. And then that free drug could eventually lead to its target. And subsequently, you can have the recycling of the receptor, and then the same thing can happen over and over again. So that's quite unique. Now, this is the specific biotin linker Basically, what's shown in orange is the paclitaxel derivative, SBT1214. And then the key functionality is having this disulfide linker. And then, of course, shown here in blue is the biotin. So this is the mechanism by which what happens over here in the endosome. So the glutathione, the sulfur of the glutathione is going to basically attack this sulfur. What's going to happen is the sulfur electrons are going to basically be pushed onto this oxygen. And I'm going to get out my pen here because they didn't show fully what's going to happen. So electrons move up. You imagine now you have some sort of, negative charge here. You have the taxoid, going to name it as tax. And then you have R, which is the resulting, part of the molecule. Electrons can swing down, and then you can kick off the taxoid like so. Sorry again if my drawing skills are not the best, but basically it's similar to the mechanisms that we saw, right? Where you're going to have an attack and then you're going to kick off something. And as a result, these are the products of the reaction. You have the free taxoid or the free paclitaxel, which can do its thing. You have the formation of this compound known as a thiolactone. And then finally, you have the biotin that's directly linked now to the glutathione. And that's basically the mechanism step by which this occurs. And I think it's, again, another unique example of utilizing chemistry to really exploiting the biological system to really help improve, treatments and whatnot. So that brings us to the end. There's a lot that we went over today, a lot of stuff that I discussed. So for instance, hopefully I've shown that mechanisms help to illustrate the elegance and the beauty of all of these, select examples of reactions, both outside and inside the biological framework. And we could build our understanding of mechanisms, as I've discussed in the earlier slides, through reviewing both general chemistry and organic chemistry concepts, like the following. Now, building a strong foundation helps us to tie visual and logical cues that I've illustrated all throughout this presentation into rationalizing chemical phenomena. And these are just common examples that that I explored here in part 1. And then, what we recently discussed is how you can exploit these enzymatic mechanisms to build novel therapeutics. And these were the examples that we covered here. And then, whenever this video or the stream is available, I would highly recommend you guys to rewatch and kind of follow what's going on, the logic behind it. Also highly recommend for you guys, if you feel free, to practice the concepts shown and to really understand how and why stuff happens. And then the last point is basically the repeat of the previous point, which is finally conceptualize, don't memorize. And that's very, very important because, at least from my experience being an undergraduate student, I realized that sometimes, so much is thrown at you, so much information is thrown at you to the point where you can't do nothing but memorize. But if you take the time to really digest it, no pun intended, if you take it, process it really, at a comfortable pace and really think about it and practice it a lot, hopefully it'll become much clearer and things will be able to be easier to recall. And usually through problem solving, that's been able to stick in my head more than just simple memorization, especially for people who are interested in becoming scientists. People want to have good problem solvers, and the way you only could be a really good problem solver is to conceptualize and not memorize. With that being said, thank you for coming to my TED Talk. I mean, my Biocord talk, and I'm now going to take some questions. So thank you very much. Okay, let's see. Where are some of the questions?
Hello, do you hear me?
Yeah, sure. Common Lewis was talking, right?
I just had a question about the spinosyn A insecticide. Insecticide. Just
Yeah,
wondering what its mechanism is. A competitive inhibitor? How does it work?
that's a good question. That's a good question. I don't think it inhibits. It's just really, at least from my understanding from just reading the paper, it just adapts that specific conformation through the hydrogen bonding of those specific amino acid residues, and then based on the established conformer, that's gonna, the bonds are aligned in a perfect way so that you can actually easily form that like 6-membered ring. So, but I don't know. I'll look into it, and if it is, I'll get back to you on that. It's a good question.
It's definitely a very specific structure. I imagine it'd be a very specific binding agent.
Yeah, it definitely looks like the case, and that's the thing when it comes to Diels-Alder. You know, you want to have a very specific conformation. I'm pretty sure it's like, there's probably like high entropy that's involved in order to really have the reaction go. That's why, for instance, you need high heat to basically get over the, activation barrier and whatnot. But I imagine through hydrogen bond interactions and other things that are occurring within like the active, that pocket within the enzyme, it's probably a little bit easier to catalyze that reaction.
Oh, that makes sense. Thank you.
Yeah, no, absolutely. Any other questions? Feel free to ask me. I'm more than happy to answer them or even help out. Oh, and by the way, if you guys want to message me with any specific questions, even if it's concepts that you had a little bit difficulty with, I'm more than happy to help out with that. I'm more than happy. Thank you for listening, Lolly. Appreciate it. Thank you, guys. Are there any other questions? I'm still going to be here, so. Or any critiques. I'm always trying to improve my presentation.
Seems fine. Maybe you could have practiced it a third time.
Third time. This guy. You're right. You're right. Definitely.
It was fun the way it was.
Thank you.
Yeah, it's
Thank you,
just a
Kraken.
joke.
I know.
Plug the server. Plug the server.
Oh, I mean, yeah. I mean,
hey, man, do you have like a server that you're on? Like, do you have anywhere where you talk about this all the time?
Yeah, I
Or
mentioned
kinda
that in
promote
like
your
the
stuff
very
like this?
beginning. There's like an organic chemistry server. I'll talk probably with the admins and see if we could like do some, I don't know, some sort of partnership or something like that. But of course, it's not only, I didn't do the talk only just for that. I really wanted to talk about the stuff because I just love enzymatic mechanisms. I just love mechanisms in general. That's like one of the things that always fascinates me about chemistry, no matter what the context is. So yeah. What are
Hi.
Hi again. Yeah, you're funny. Okay. Sure. 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 That sounds great. Thank you so much for setting this all up. And you guys did a phenomenal job of just moderating everything and even recording. Like, it's really, really impressive. So good job on that.